Our second major grouping of proteins in terms of tertiary structure is globular proteins. As we will see later, globular proteins do much of the work of the cell in terms of enzymatic activity. In contrast to fibrous proteins, globular proteins have backbone chains that do fold back on each other. Interactions between side chains play a key role in adopting the final tertiary fold. We'll look at the structure and function of myoglobin, pictured here, in some detail in a later lesson. The major factor driving the formation of protein structure is the hydrophobic effect. Recall that polar functional groups readily associate with the polar structure of water, but nonpolar groups tend to be excluded by water. In the formation of a globular protein, we find that most of the polar side chains are on the outside on the water-exposed surface. The types of interactions with water include hydrogen bonds and ion-dipole interactions. In contrast, nonpolar side chains tend to be buried on the inside of the protein sphere. We can see this illustrated here, where the hydrophobic core is illustrated by the green spheres on the left and the red spheres on the right are the hydrophilic surface. Globular proteins have substantial sections of alpha helices and beta strands. As is true for myoglobin pictured here, some are entirely alpha helical in secondary structure. Globular proteins tend to be soluble in water and salt solutions, thanks to their hydrophilic surfaces. They often have very compact structures, in contrast to the extended structure of fibrous proteins. Let's see what kind of non-covalent interactions help define tertiary structure. Note that these examples apply to either fibrous or globular proteins. Hydrogen bonds might be present between polar side chains, and nonpolar side chains can form hydrophobic interactions. There may also be electrostatic attraction between side chains carrying opposite charges. In some cases, a metal ion is crucial to protein function and is often coordinated to side chain residues. As we've seen in another lesson, disulfide bonds between cysteine residues are strong covalent bonds that can restrict the folding patterns of proteins. The primary lesson here is that the final conformation of a protein is the result of the interplay of all the stabilizing forces.